So given the title of the seminar series, obviously I'm sure everyone here is familiar with the idea that there's at least two kind of parallel visual systems, in addition to the conventional visual pathway, which takes in the visual thalamus and superior colliculus and supports our detection and response to form and motion in the world around us. We've got this so-called non-image forming system, which involves bits of the pretectum, thalamus, and particularly hypothalamus, that regulate subconscious responses to light, particularly things like the pupil light reflex and regulating circadian rhythms, etc. So although obviously we've known about this for many, many years, I think the idea of a non-image forming system really came from the discovery of melanopsin and the iPRGCs, and the realization, or at least what it seemed at the time, that they selectively projected to these bits of the brain, regulating subconscious responses to light, and of course also that the properties of those intrinsically photosensitive ganglion cells seem to be specialized to encode changes in ambient illumination. So they have sustained responses to maintain changes in light levels, and those come via their both a combination of their intrinsic melanopsin-derived response and also signals coming from the rods and cones in the retina. So obviously we've known about this for about 25 years or so now, but over the years, I think the distinction between conventional and non-image forming visual systems has blurred a bit. On one hand, because 10, 15 years ago, we learned that actually iPRGCs are more numerous than we originally thought, and they also project to bits of the brain involved in conventional aspects of vision. In mouse, the dorsal lateral geniculate nuclei, and they contribute to thalamocortical visual processing and aspects of vision. Since these original studies, there have been many studies showing roles of melanopsin in animal and human vision. On the flip side, though, we've also learned that what's going on in the non-image forming system is a bit more sophisticated, perhaps, than originally imagined. So, for example, our group has showed that cells in the master clock in the hypothalamus, the suprachiasmatic nucleus, some of those cells can respond to spatial patterns like gratings, and there's also many cells in the SCN that seem to be able to detect color, and we've shown that those color signals regulate both circadian behavioral responses and also acute behavioral responses to light, such as masking and light avoidance or aversion. What I'm going to talk about today, I think, even more greatly blurs the distinction between vision and non-vision. So I'm quite excited about this stuff. Before I get to that, just one point I'm going to make, which is that a key tool that we've used to provide insight into the photoreceptive pathways regulating both vision and non-vision is this mouse line developed about 20 years ago or so by Jeremy Nathan's group, where the native M-cone opsin in the mouse is replaced by the human L-cone opsin. And that shift in cone spectral sensitivity gives us a neat sort of handle where, by using lighting with different spectral composition, we can selectively modulate brightness for one or more photoreceptors of our choosing, and then figure out how the different opsins are contributing to the responses. So I mentioned that here just because we've used this mouse line for some of the experiments I'm going to talk about today. But as I say, I think really it goes far beyond just the photoreceptive signals to the more conventional aspects of vision that we've been studying. So what I'm going to talk about, so obviously much of our understanding of the non-visual system, certainly in terms of hypothalamic light responses, has come from studying one very specific part of the hypothalamus, the suprachiasmatic nucleus. But that's not the only bit of the hypothalamus that gets retinal input. And so for a while now, we've been interested in another lateral branch of the retinohypothalamic tract, which projects to the lateral hypothalamic area. So you can see it here on the image on the left. So the subset of retinal ganglion cells project to the lateral hypothalamic area. They're particularly dense in the sort of ventral parts, which is often known as the perisupraoptic nucleus, because it sits just above the supraoptic nucleus. But you can also see axons going right up into the lateral hypothalamic area proper. So this is a visual pathway that's highly conserved across mammals. I think it's been found in every mammal that's been investigated, including humans. And we know it's an input that drives at least partly via a particular type of intrinsically photosensitive retinal ganglion cells, the M1 type. So that's actually what's shown here in the image. This is a transgenic line that selectively labels the M1 IPIGCs. But beyond that, we really know nothing about the function of this pathway. So that's what we set out to look at. That's what I'm going to talk about today. So we started by simply surveying activity across the hypothalamus, the lateral parts of the hypothalamus, looking for light responsive neurons. So on the left here, you can see examples of two of those recordings. So we've got a linear 30-site multi-electrode inserted into lateral parts of the hypothalamus. This is in anesthetized mice with those red cones I mentioned before. And the panels to the side show you the activity, change in population activity evoked by a 2-second light pulse, quite bright, short wavelength light applied to the contralateral eye. And so what you can see is that at most of those electrode sites, each line is an electrode site, nothing happens at all. But there's a discrete sort of zones within these. So in this recording, it's this set of sites just down near the bottom. In this slightly more lateral recording, it's a set of sites actually more dorsal within the brain. You can see clear and robust light evoked activity. So we did a bunch of those recordings. This is a sort of heat map showing the average change in population firing across 68 recordings spanning bits of the hypothalamus. And you can see a very discrete region starting at the kind of ventral just perisuperoptic region and extending laterally and dorsally up into the lateral hypothalamic area where there's very robust light evoked activity. So that's not particularly surprising given what I just showed you, what we know about the retinal inputs. What was more surprising to us was that if you look at the responses that we get, you can see often there's a burst of firing when the light goes on, and that decays within a second or so, and then another burst of firing when the light goes off. But this is really different from what you might generally expect of the hypothalamus, right? There's minimal evidence that cells here are encoding steady state light levels, in which case you'd expect firing to remain high throughout the duration of a light step. So you see that occasionally, but not as a rule. So to look into that in more detail, we isolated the activity of individual cells that were detected in these recordings. And here's the average profile of responses from cells, and we found we could split them into two groups. So in fact, there were some cells that showed somewhat sustained increases in firing response to these 2-second bright light steps, but a greater number of cells, so more than 50% of cells, showed these transient increases in firing at light on and light off, but had no sustained activity at all. So these cells clearly cannot be encoding irradiance, which is the sort of prototypical signal you would expect hypothalamic neurons to detect. And just to hammer that home, we did an identical set of recordings from the SCN, suprachiasmatic nucleus, and here you can see the vast majority of cells show very sustained increases in firing in response to the same stimulus, and very few cells show this transient firing. And in fact, these are mainly cells located just outside the SCN. This is obviously all done so far in those mice with the red cone knock-in that I mentioned. I'll get back to how we've used those in a moment. Just at this point, I just want to make the point that we see something very similar if we record also from the SCN from the lateral hypothalamic area of wild type animals. So essentially exactly the same thing. So you'll note that I mentioned the stimuli we're delivering here is a bright light step applied to the contralateral eye. In these recordings from wild type mice, we also asked whether maybe it's something to do with the fact that we're just stimulating the contralateral eye, which is why we don't see very sustained responses. In fact, however, we found no cells that responded exclusively to stimulation of the ipsilateral eye, that's shown here in blue, and only a very small minority that showed any ipsilateral evoked activity whatsoever. So some kind of binocular convergence, but very weak. So almost all of the cells in the lateral hypothalamic area receive their input from the contralateral eye. And again, that's completely different from what you see in the SEN, where you see large numbers of cells that show exclusively ipsilateral driven responses and large number of cells that show responses to stimulation of both eyes. So there's some sort of fundamental difference in the types of light responses that you see in the lateral hypothalamic area versus the SCN. So we went on to look at, in a bit more detail, at the photoreceptive pathways that are driving these responses. So this is our data from the red comb mice. And so what we did here to start with was we applied short and longer wavelength light steps, 460 nanometers and 655 nanometers. We matched the brightness of those two stimuli, so they should appear of identical brightness to the L cones in these mice. And we presented them over 7 different orders of magnitude in brightness. So the top one here is the stimuli I've just showed you for the blue. And so this is an example cell on the left. These panels here show the mean population responses for the acute change in firing during the first 250 milliseconds, and for later components of response, the last 500 milliseconds. So this is for the cells that I showed you before that have the sustained firing in the lateral hypothalamic area, so the less commonly observed cells. And what we found for those is that this initial transient increase in firing, sensitivity to the short and longer wavelength light was pretty similar, and overall sensitivity was quite low. So we only start to see significant responses once we get up to relatively high light levels. So both of those things coming together suggest that this acute response of those cells is primarily driven by cones. And that's similar actually to what we see in the SEN as well, with a similar stimuli. And then looking down here at the more tonic components of the response, you'll see again the response to the two wavelengths is pretty similar, but it starts to diverge a little bit when we get up to high light levels, which would be consistent with a melanopsin contribution to those responses. So obviously, melanopsin is far more strongly activated by the shorter wavelength light. I'll come back to that point in a moment. For now, really, the main point I wanted to make is this is very, very different from what you see when you look at the transient cells within the LHA. So again, here's an example neuron, and here's the population data. So you can see that actually responses to the short wavelength light, if we just focus on the initial acute on excitation of these cells, they tend to be bigger for the short wavelength than the longer wavelength, particularly at lower intensities. So this is strong evidence that actually there's a substantive rod component to the response of these transient cells that you don't see in the sustained cell population. And as I mentioned, you don't see that in the SEN either. That's even more pronounced if we look at the off excitation of these cells. You can see that for the short wavelengths, they respond even very robustly, even at the dimmest light we tested. And indeed, if we correct the x-axis on these graphs, so now rather than looking at L-cone effective photons, we're looking at rod effective photons. You can see that the responses at these lower intensities align with what you would expect for rods. So the transient cells are much more sensitive than the sustained cells, and that comes from a more substantive rod input. As I mentioned, the other thing that seems to differ between these is obviously, whereas these guys have this sustained firing that shows evidence of a melanopsin component, it's lacking in the transient cells. We wondered whether maybe something about the stimuli we were using for these experiments was kind of giving us a slightly misleading picture, maybe, of the extent to which there was sustained melanopsin-driven responses to these cells, because these are quite short stimuli, and melanopsin's well known to be quite sluggish in terms of its light responses. So in an independent data set, in this case, collected with high-density electrodes targeting specifically the retina recipient regions of the lateral hypothalamus, we started using a different set of stimuli now. So we still have our short wavelength 460 nanometer light. Now our longer wavelength stimuli, we've added in a tiny bit of violet. It's quite hard to see on this graph, but together that means that the brightness of these two stimuli is matched for both L and S cones. And we present longer and brighter light stimuli here compared to what I've just shown you. And sure enough, when we do that, we can again separate cells into a sustained and transient population. And what we find is the sustained population reliably show bigger responses to the melanopsin high versus melanopsin low stimulus, particularly for more sustained components of response. Here we're comparing the initial on excitation to the response 5 to 10 seconds after the light appeared. And the transient cells respond identically to both stimuli. That's good evidence then that the sustained but not transient cells are getting melanopsin input, which is what you would expect. Just to further validate our stimuli here, we repeated the experiments in red cone mice where the melanopsin gene was knocked out, so red cone melanopsin knockouts. And in those guys, again, you can find sustained and transient cells, with the transient cells being more numerous. But here, importantly, in neither case do you see a difference between the blue melanopsin high and the longer wavelength melanopsin low stimuli. So putting all that together then, there's clear evidence that these two cell populations differ in terms of their input. The transient cells have more rod input, the sustained cells have melanopsin input. And both types probably get cone input. To look at the cone inputs in a bit more detail, we then used a next set of stimuli. So there's actually 4 different stimuli here. They're polychromatic stimuli. And we're presenting pairs of stimuli that are matched to have identical brightness for most opsins, but selectively different brightness for either just L cone opsin, just S cone opsin, or L plus S cone opsin, so a sort of uniform increase in brightness specifically for cones, or this last one, which is an L minus S stimuli, where the net brightness for cones stays the same, but there's a change in the relative balance of L versus S cones. So it's a chromatic blue-yellow type modulation by analogy to the human visual system. So using those, we were able to find 4 different types of responses. So a bunch of cells that show either they only respond to just one of the 2 cones, so they either have L-driven responses or S-driven responses, or if they respond to both, they respond in the same manner. So those inputs are additive and they respond strongly to L plus S luminance modulations and more weakly to L minus S chromatic modulations. But we also see groups of cells with clear color opponent responses, either L on, S off, or the converse. So much as we've reported previously for the SCN and indeed many other brain regions, there's plenty of cells here in the lateral hypothalamus that seem to be color opponent. This graph here just shows you the relative response to L versus S cone stimuli. So there's just an index where -1 means they're purely S cone driven, 1 means they're purely L cone driven. And you can see the non-opponent cells tend to be very strongly biased. So some are very strongly S cone driven, some are very strongly L cone driven. The opponent cells are more kind of equal in their preference. And across the sustained and transient cells, we find numerous examples of color opponent cells in both groups. So there seems to be no kind of no clear relationship between whether they're sustained or transient and whether they have color opponency or not. So just to summarize that first part, I've shown you that contralateral retinal signals drive substantial light-evoked activity across the rostral lateral hypothalamic area, and therein we find 2 broad classes of visually responsive neurons. On one hand, cells that show relatively low sensitivity, sustained irradiance coding responses, similar to what you'd see in the SCN, and their responses are primarily driven by cone and melanopsin input. On the other hand, a greater number of cells that show much higher sensitivity, and they respond to visual contrast, but they don't encode irradiance, and their responses are primarily driven by rod and cone input. both types of those cells display a diversity of cone inputs in terms of whether they're coming from L and S cones, and that includes many cells that exhibit color opponents. Okay, so that sort of is interesting in the first part, but it prompted us to ask, well, where is the input to these cells coming from? How do they respond to stimuli that are more than just uniform changes in light level or color? So to start looking at that, we used a monitor positioned either directly in front of the mouse or moved slightly laterally. So we're mainly covering kind of frontal and upper parts of the visual field. For reference here, this red cross indicates the retinal midpoint. And we started by simply presenting flashing bars, either black bars on a white background or white bars on a black background, either horizontally or vertically. And we asked how the cells responded to that. And what we found was that actually very many cells showed very, very localized responses. So they only responded to bars in a very particular part of the visual scene. So just to orientate you to these plots at the bottom, this is 6 different example cells, either recorded when the monitor was positioned like this, these 3, or for the 3 on the right when the monitor was positioned laterally. So the top panels show the response to a vertical bar as a function of where it appears on the azimuth plane, and the bottom panels show you the response to a horizontal bar based on where it responds on the elevation plane. And what you can see then is these are the response to a white minus black bar. So white regions means the cell was firing more when it was light versus dark, and vice versa. So you can see very discrete receptive fields for these cells occupying very narrow region of visual space and very often surrounded by an antagonistic surround. So for example, this cell here liked a light bar, and it increased firing for a light bar there or a dark bar above or below it. So we could then ask where in visual space these cells get input from. We found that the receptive fields were very strongly clustered in this kind of central region here, which corresponds to just in front of the animal and kind of just above just above it. So it's broadly speaking superior and temporal regions of the retina that those signals are coming from. What was particularly interesting, though, was not only is it clear from this that there seems to be a non-random distribution of where the inputs to the lateral hypothalamus are coming from in visual space, but also just that almost all of the neurons we recorded responded to these. So that was true of 75%, 80% of the transient and sustained cells we recorded, but it was also true of 75% of the cells we recorded that actually didn't respond at all to full field stimuli. So some of these cells are incredibly selective for spatially restricted stimuli, and they don't respond to uniform changes in light levels at all. Interestingly, we also found that there was some kind of anatomical order to where the cell's receptive fields were, or some relationship between where in the LHA the cells were located and where their receptive fields were in visual space. So in particular, what you can see here is that there was a trend so that cells in the more dorsal parts of the lateral hypothalamic area tended to have receptive fields at higher elevations than cells in more ventral parts of the lateral hypothalamic area. There was no detectable variation on the medial to lateral axis. But this is sort of somewhat resembling what you might see in more conventional visual areas, where there's obviously a very strong retinotomic order to retinal projections, but it's pretty crude in comparison. We did wonder whether perhaps the fact that we're only measuring visual signals from a relatively restricted part of the visual field might limit our ability to observe a retinotopic order here or skew our results. So to address that in a slightly different way, we also took data that I showed you before, where we assessed the preference of cells to L versus S cones. And that's kind of potentially informative because, as many of you will probably be aware, in the mouse retina, there's a strong gradient in the expression of S versus M cone opsins, with the ventral parts of the retina strongly biased towards the S opsin and more dorsal parts of the retina strongly biased towards the L opsin. So our prediction based on these data is that that variation in cone preference should be mapped anatomically within the lateral hypothalamic area. And indeed, that's what we found. So we found more dorsal parts of the lateral hypothalamic area was strongly biased towards S cones, corresponding to receptive fields at higher elevations, and more ventral parts strongly biased towards L cones. So there's a crude retinotopic order to visual input to the lateral hypothalamic area, somewhat analogous to what you might see in more conventional regions of the brain. Moving on, we also then, given that these cells are strongly selective for spatial stimuli, we next asked, well, perhaps they might also be sensitive to motion. So to test this, we applied a set of drifting gratings of varying speeds and size and moving in different directions of motion, as you can see here. And we found, again, a great number of the cells we recorded respond to these stimuli, even more than we could map a receptive field for, in fact. So it was only a tiny proportion of the cells we recorded overall that didn't show detectable responses to these stimuli. And in many cases, what we saw was something like these two example neurons shown on the bottom left. So here you're looking at the average response across one cycle of the grating. You can see these cells show robust modulations in firing rate in response to the grating, and they respond similarly to all the different tested directions of motion. But we also found some cells that seem to be very selective for the direction of motion. So here's an example of two neurons that strongly responded to stimuli moving in the nasal or nasal dorsal direction. Similarly, we also found other cells that seem to be strongly tuned to orientation. So, for example, here's a cell that likes gratings kind of moving diagonally in either direction. Here's a cell that likes gratings moving kind of up or down, but not horizontally. Here's the summary plot for all of the cells we tested here. So cells were defined as direction or orientation selective using established indices. We found a small but substantial, nonetheless somewhat substantial, or at least surprisingly substantial subset of cells that were tuned to detect either directions or orientations of motion. The direction selective cells, as you can see here, were most commonly either cells that had transient responses to full field light steps or didn't respond to full field light steps at all. The orientation selective cells included some more of the cells that showed sustained responses as well. So we next went on to ask, given that there are some cells that seem to respond to simple moving stimuli, maybe there's also cells in the lateral hypothalamic area that respond to more complex motion. So particularly here, we were interested in responses to approaching and receding motion, since this is a potentially sort of behaviorally salient stimulus. For example, some of you will be aware that it's well known that sort of approaching or expanding dark discs evoke robust kind of defensive behavioral responses to rodents because they're thought to simulate overhead aerial predators. So we tested those kind of stimuli. So just to orientate you, what we tested here were either dark spots that got bigger, dark spots that got smaller, light spots that got bigger, or light spots that got smaller. And to kind of confirm that any responses to those were due to the motion rather than just overall changes in light levels, each of these was paired with a control stimuli where we had a spot that remained of a fixed size but changed in brightness. So again, we found lots of cells responded to this. Most of the cells responded as you might expect a cell that detects the luminance of a particular part in visual space to respond. So, for example, they either kind of were excited by either a dark spot getting smaller or a light spot getting bigger, and inhibited by a dark spot getting bigger or a light spot getting smaller. So an on-type response or the converse off-type response. But we also found cells that selectively responded to the stimuli that changed in size versus the matched luminance controls. For example, here are cells that selectively respond to either a light spot or a dark spot getting bigger. Here are cells that respond to either a light spot or a dark spot getting smaller. And here are cells that respond to either light or dark spots getting bigger or smaller. So we call those respectively lume sensitive, recede sensitive, and lume and recede sensitive neurons. And overall, that was about almost a quarter of the cells that we detected showed one or more of those types of motion sensitivity. So really, this goes far beyond what you would have expected for the hypothalamus. Rather than simply detecting changes in brightness, we've got cells that are sensitive to spatial contrast and or motion. And indeed, just to summarize that, overall, we found 35% or so of the cells we recorded showed some evidence of motion selectivity, either to the drifting gratings or to those approaching spots I just told you. And half of those cells didn't respond at all to uniform changes in light levels. A further 20% of cells were specifically selected for spatial contrast, so they didn't have motion selectivity that we could detect, but they showed a detectable receptive field, but no response to uniform light. So overall, it seemed to be like more than a third of the cells we recorded in the lateral hypothalamus are completely insensitive to uniform light, but they do respond to some type of spatially structured stimuli. So there's a hell of a lot of diversity going on here in the lateral hypothalamus in terms of the types of visual signals cells will respond to and prefer. Further told you that most of the cells in this region have clear center surround receptive fields, which explains why many of them don't respond to uniform stimuli. And that the visual responses in this region are primarily deriving from the frontal and upper visual field, with there being a crude retinotopic order, it seems, to the visual responses in this region, where more ventral locations are represented more dorsally in the lateral hypothalamic area. Okay, so based on those data, we went on to look at retinal projections to this region to sort of confirm some predictions that we might make. So one is that they should be non-uniformly distributed in the retina, the RGCs that project to the lateral hypothalamic area. So to do that, we injected a retrovirus into, or a virus optimized for retrograde transfection into a reporter line. So the virus delivers the enzyme pre-recombinase that switches on the reporter in these AI32 mice. And then we can identify the retinal ganglion cells that project to the lateral hypothalamic area based on EYFP expression. And here we've co-stained the retinas with S-conopsin. So as you can see, we have this pronounced gradient in the expression of S-conopsin with much stronger density of S-cone expressing cells in ventral parts of the retina. And that aligns also with where we find more dense expression of the EYFP tag that marks the LHA projecting RGCs. So we find a non-random distribution of LHA projecting retinal ganglion cells with the highest numbers in the ventral temporal quadrant of the retina, contralateral retina, I should say, which exactly matches what we saw in the electrophysiological recordings. Similarly, if we look in the ipsilateral retina, we see very few retro-labeled RGCs, and those are exclusively found in the ventrotemporal part of the retina. We next went on to look at the identities of these retinal ganglion cells in a bit more detail. So obviously a clear prediction of the electrophysiological data I just showed you is that many of these ganglion cells that are projecting to the lateral hypothalamic area should lack melanopsin expression, because many of the cells we recorded in the LHA showed no evidence of melanopsin input. And indeed, that was what we found. So we were able to find about half of the cells, in fact, we retro-labeled showed detectable melanopsin expression. So here's an example here. They have a sort of morphology that resembles the M1 type RGCs that are known to project to this region, but we also found a great many cells that lacked detectable melanopsin immunoreactivity that we could retro-label from the lateral hypothalamus. And these seem to show a diversity of different morphologies, but often with quite small, densely branched arbors, in some cases being asymmetric. So obviously the lateral hypothalamus, where we're performing these injections, is quite close to the optic tract. So one possible concern is that we're just non-specifically labeling some retinal ganglion cells. We're pretty confident that isn't the case, because if we do the same approach injecting into the SCN, which is even closer to the optic tract, as expected, almost all of the cells we retro-label are melanopsin immunoreactive and have a morphology consistent with the M1 type cells that are known to project to that region. So these plots at the bottom just show some quantification of that in terms of the dendritic field diameter and soma size for cells that we could retro-label. And this just highlights the fact that for the non-melanopsin immunoreactive group that project to the lateral hypothalamic area, shown in red, there's a diversity of dendritic field diameters and soma size, suggesting at least two different populations of cells. Based on another study which suggested that some of the retinal ganglion cells that project to the lateral hypothalamic area might be direction selective ganglion cells, which would of course align with some of the data I just showed you on motion selectivity. We asked whether these LHA projecting cells expressed CART, which is a known marker of direction selective retinal ganglion cells. We were able to find occasional examples of cells that did express CART, but most cells were CART negative, and where we co-stained with melanopsin, there were also, the melanopsin negative cells were very often CART negative. So this suggests actually there's, at minimum 3 different types of retinal ganglion cells that project to the lateral hypothalamic area, probably more. We next wanted to get some more insight into the retinorecipient neurons in the lateral hypothalamic area themselves. Where do they project to? What type of cell are they? So to look at that, we used a different viral approach. We injected a virus into the retina that to express Cre recombinase, and this AAV1 serotype is known to show some transsynaptic antegrade spread. And then we injected a Cre-dependent reporter protein into the lateral hypothalamic area. And sure enough, we're able to antegradally label a set of neurons across the lateral hypothalamic area, matching the kind of places where we found light responses. Just some examples shown here. And where the neat thing about this approach is, because of the reporter we used, which labels the process of the cells, we could trace where they project to. So this is just the tracing of a bunch of cells where we had the most, from a bunch of animals where we had the most robust labeling. And we found projections not only across the hypothalamus, but also to a number of extra hypothalamic sites, including the septal complex, habenula, and hippocampus. So just some images there of the labeling we saw. We also then took the opportunity to ask what type of signals are these providing? Are they glutamatergic or GABAergic? So to do this, we co-stained alternate sections with markers of glutamatergic or GABAergic neurons, so VGLUT2 for glutamate, VGAT for GABA. And then using confocal imaging, we looked for co-localized voxels. So to control for kind of spurious co-localization here, what we do is we look in small subfields, we quantify the number of voxels that co-localize M-cherry and the synaptic marker, and then we rotate the synaptic marker channel and look at the sort of co-localization that might occur by chance. And to cut a long story short, when we did that, we found that in every region of interest examined, we saw significant co-localization with the glutamatergic VGLUT2 marker and never with the GABAergic VGAT marker. Consistent with those data, we also did some experiments where we used optogenetic stimulation to identify GABAergic neurons. This is a mouse line where GABAergic neurons express channel rhodopsin 2. And what we found was that among the visually responsive neurons we recorded, while we could find a number of cells that showed inhibitory responses to optogenetic stimulation, so a decrease in firing when the light was on, followed by a rebound excitation when the light went off, consistent with GABAergic input to those neurons, no cells showed the direct excitatory response we would expect for GABAergic neurons. And importantly, this included the full spectrum of different response types I told you about that we found in the red cone mice. If we moved our electrode to adjacent non-light responsive regions, we could very readily identify cells that were directly excited by our light step. So we're confident the optogenetic approach worked to identify GABA cells. It's just that these light responsive ones are not GABAergic, they're glutamatergic. Just to summarize that before I get to the final part, I've told you that the lateral hypothalamus projecting RGCs preferentially localized to ventral temporal retina. And the lateral hypothalamic area receives input from multiple ganglion cell classes. So about half of them seem to be melanopsin-like M1 type cells, and more than one class of melanopsin negative RGC project there. And then I've also told you that the retinal recipient hypothalamic neurons project widely up to extra hypothalamic sites, including the septal complex, habenula, and hippocampus, where they seem to drive glutamatergic-based responses. So the final little bit then, we wanted to understand the function of this pathway in more detail. So to do this, we selectively expressed DREAD receptors in retinal ganglion cells that project to the LHA by combining an AAV retro Cree injected into the LHA with a Cree-dependent DREAD virus injected into the retina. So just to orientate you, we either used excitatory GQ coupled DREADs or inhibitory GI coupled DREADs, or just a control vector. And in the first instance, we took animals in constant darkness and then activated the DREAD receptor with IP injection of a ligand, low dose clozapine, around the mid-projected day. And then we looked for initially CFOS expression in the retinal cells that projected, in the retinal cells that are labeled by this approach. So what you can see here then is when the animals had the GQ DREAD, many of those LHA projecting RGCs labeled in red also expressed CFOS, showing they were activated. We saw only very few activated neurons in either the control or the GI DREAD animals. So this confirms that we're able to manipulate the activity of the retinal ganglion cells that project to the lateral hypothalamus. We also found when we looked in the lateral hypothalamus itself that there was significantly more CFOS expression in the GQ DREAD expressing group than in the GI DREAD group, as you would expect. So we can definitely modulate retinal input to this region. And those findings also, just to make the point, we didn't see any difference in the other major retinal recipient part of the hypothalamus, the SCN. But importantly, those findings also held true for the target sites I mentioned before, at least some of them. So we found a significant increase in CFOS expression in both the septal complex and habenula in the GQ DREAD versus GI DREAD animals. We didn't see a difference in the hippocampus, where there was virtually no CFOS expression. So the final bit of data, clearly this approach allows us to modulate activity of this circuit. What's the circuit actually doing in terms of behavior? So to look at that, we took animals, those DREAD expressing animals, we housed them in an arena where they were exposed to various visual or non-visual stimuli. So either an expanding disc, excuse me, kind of a looming type stimuli, a simple light flash, or an auditory stimulus. And we used a kind of markerless 3D pose estimation based on deep lab cut here to look at the mouse behavior in higher dimensions. So from that, we could extract not just information about locomotion, but also rearing, whether they're rotating, bending their body, etc. So first of all, here you're looking at some data for when the animals received an auditory stimulus. So the auditory stimulus comes on at time naught. And you can see that essentially the behavioral response is a kind of brief increase in locomotion, body bending, some rotation. So the animal's kind of looking around, what was that noise? But importantly, the response was very similar in animals that had either the GQ, GI dread, or control. When we then looked at a visual stimuli, so this is now an overhead looming disc, you can see a very strong response where animals tend to freeze, reduce locomotion, and rear. And we found that components of that response were selectively increased in the GI dread animals. So more freezing, more decrease in locomotion. So this is consistent with the idea then that actually what retinal input to this region is normally doing that we've switched off here is opposing that freezing response to the overhead looming disc, presumably because it's driving an alternate behavior, such as running away. And consistent with that, when we looked at response to a simple light flash, we found light flash drives increases in locomotion, and that response was reduced in the GI dread and in fact in the GQ dread animals, presumably because either increasing or decreasing the activity of these retinal neurons abolishes the behavioral response to, or abolishes the ability of the animal to detect that change in light levels. So just to summarize then, I've told you that visual responses are surprisingly diverse and widespread across the rostral lateral hypothalamic area, with many cells selected for spatial temporal contrast. The lateral hypothalamic area receives input from multiple retinal ganglion cell classes, and the retinorecipient neurons therein send glutamatergic projections to brain regions involved in behavioral control. And I've then finally told you that manipulation of this retinal input alters behavioral responses to visual stimuli, which is consistent with a role in these inputs in driving escape behaviors to specific types of visual stimuli. So obviously, this is probably only scratching the surface of what this system is before, given the diverse kind of visual responses. Perhaps there's a wider role for this system in other types of visually guided behaviors, such as hunting and or effects of light on memory, mood, and cognition, etc. So I'm going to end there just by thanking the people that were involved in the work. So particularly Josh Muland and Elizabeth Tamayo did most of the experiments, valuable contributions from many other colleagues here at Manchester, and particularly the overall time, vision, and behavior grouping in our Centre for Biological Timing. So I'm going to end there. Thanks.